Good morning, students. This is Dr. Ankita, Assistant Professor from Department of Microbiology. So the topic that I'll be teaching you today is sterilization. And as you all know, sterilization is a very important topic in microbiology syllabus, and it is useful both in theory as well as in practicals. In practicals also you'll get it in some form in Viva. There can be spotting also, and for theory there will be a definite question from sterilization chapter. So this has to be read very sincerely. So moving on with our slide, the lesson plan for this presentation would be that it would include the various definitions. We all use the terms differently, like we use sterilization, then what is sterilization, what is disinfection, what is antisepsis, asepsis. So these terms, they appear to be similar, but all are different. So that I'll be discussing, and then there'll be factors that influence the efficacy, then what were the agents of sterilization, and then we'll study autoclave and hot air oven in detail. Okay, and then we'll also discuss about filtration and some part from radiation also. So moving on with the definition of sterilization. So the definition should be learnt by heart, and try to divide your definition in different parts. You try to divide it in different bullets, and then you make a proper definition of it. So how can you define sterilization? It is the process by which all living microorganisms, including viable spores, they are either destroyed or removed from an article, surface or medium. So now you all know what are spores. Spores are the resistant stage of bacteria. Okay, so in sterilization, we are removing all the living microorganisms, including the spore form, which is the resistant form. And they are either destroyed and or removed from an article, surface or medium. Now, if we talk in terms of numbers, then there is reduction of more than equal to 10 to the power 6 log CFU of microorganisms. Okay, what is CFU? CFU means colony forming unit. It is assumed that one bacteria forms one colony. So there is reduction of more than equal to 10 to the power 6 log CFU of microorganisms. Okay. Now then there is another term which is disinfection. So in disinfection, we remove or destroy the pathogenic microorganisms, and bacterial spores may be reduced or may not be reduced. So not necessarily the bacterial spores are reduced. And disinfection is used on inanimate objects, non-living objects, and not on body surfaces. If disinfection is done on body surfaces, then we call it as asepsis. And the materials that we use for asepsis are known as antiseptics. So how you will define antiseptics? Antiseptics are the type of disinfectants which are safe to apply on body surfaces, on skin and mucosa, resulting in destruction of microorganisms present on body surface. So the process is known as asepsis, and materials that we use are known as antiseptics. So you all may be aware of both these terms. During the COVID pandemic, everybody has come across disinfection, antisepsis, which is the only thing that can prevent us from the pandemic, which we can take up as a precautionary measure that can prevent us from having the COVID. So that was sterilization, disinfection and antiseptics. We can revise it again. In sterilization, we are removing the microorganisms, including the spore form, from an article, surface or medium. In disinfection, we are not removing the spore form. If they are used at a higher concentration, if the disinfectants are used at a higher concentration, then maybe the bacterial spores get killed. Then it will be known as a sterilant. And when disinfection is done on living things or on animate objects, then it is known as asepsis, and the material required are known as antiseptics. Okay. So I hope the definition would be clear to all. Then what are the factors that influence the efficacy of a sterilant? So in today's lecture, I'll only be discussing about sterilization, as it is a very vast topic. So it has been divided into two, sterilization and disinfection. I'll be only discussing sterilization in my lecture today. So what are the factors that influence the efficacy of sterilant? So firstly, it is the organism load. How much organism is present in that article, surface or medium. More the organism, more powerful sterilant has to be used. Then there is nature of organism. So we all know that nature of organism includes the nature of microorganism that is present, whether it is a bacteria, whether it is a virus, whether it is a parasite, or whether it is a fungus. So I'll be discussing this in detail in my next slide. Then the third point is concentration. So the concentration at which we are using the sterilant. At a higher concentration, less time will be required and more microorganisms will be killed. And if we dilute the sterilant, then less microorganisms will be killed. Then there is a duration of exposure. For any sterilant or disinfectant, there is a certain period of time that has to be there for contact between the disinfectant sterilant with microorganisms. Then only the killing process happens. Then there is temperature. Every sterilant disinfectant have their own temperature at which they work. Local environment, which includes the pH, the biofilm formation. So in microbiology, when you'll be studying, then you will see that the microorganisms, they form a biofilm. They form a layer on the article, surface or medium, that is biofilm, and that also hinders with the action of sterilant and disinfectant. Okay. Now the nature of organism that I was discussing. So the decreasing order of resistance to sterilizing or disinfecting agents are prions. Prions, they are the highest, they have the highest resistance. Then there is bacterial spores, then mycobacteria, fungi, large non-enveloped virus, and the enveloped virus. So again, in this COVID endemic, we have seen that coronavirus is an enveloped virus, and just by merely hand washing with simple soap and water, we can get rid of coronavirus from our body surfaces. Okay, from our hands. So enveloped virus, they are very easily removed, but prions have the highest resistance toward sterilizing agents or disinfecting agents. Okay. Now the agents of sterilization and disinfection, they are mainly divided into two broad categories: physical agents and chemical agents. Physical agents include sunlight. So living in India, we all know anything we have to get dry or we have to sterilize, we keep it in sunlight. Then there is drying. We can go for drying method, then heat method, filtration method, and radiation method. So these are all physical agents. Okay. Now then comes the chemical agent. These are all physical agents. Now come the chemical agents, which include different disinfectants or different chemicals. They can be phenols, they can be halogens. You all know, you must have studied in your school days also that we use chlorinated water. So chlorine is a halogen and it is used for purification of water. Then there are metallic salts, aldehydes, alcohol, very important for disinfection. So when you are using alcohol based hand rub, then you are disinfecting. No, you are doing asepsis of your hands. You are removing the coronavirus by using a sanitizer. Some certain dyes can also be used. Then there are vapor phase disinfectants and surface active disinfectants. Okay, so the disinfectants, they are broadly classified into major two categories, either physical or chemical. So this physical part is what I am going to discuss today, and the chemical one is altogether a different lecture. Okay. So now in physical we had first of all sunlight. So in sunlight we all know there's nothing to discuss in it. कुछ भी घर में होता है we keep it in sunlight और धूप में सूख जाता है ना. So उससे we assume कि वो थोड़ा सा sterilize हो चुका है. Then drying है ना, किसी भी चीज को अगर ड्राई कर दो तो भी होता है. ऐसी बैक्टीरिया और एनी माइक्रो ऑर्गेनिज्म इट नीड्स मॉइस्चर है ना, उनको उसको मॉइस्चर चाहिए, न्यूट्रिशन चाहिए. इट ऑल्सो नीड्स मॉइस्चर फॉर ग्रोथ. तो अगर मॉइस्चर नहीं होगा तो फिर बैक्टीरिया या माइक्रो ऑर्गेनिज्म ग्रो भी नहीं करेंगे, है ना। तो किसी भी चीज को ड्राई कर दो, इट विल नॉट ग्रो. यू टेक इट इन पाउडर फॉर्म, यू ड्राई इट, it will not, the microorganism will not grow. So that is also a part of sterilization. Then the third part of sterilization, that was heat sterilization. This one is important, and this classification is also very important. In heat sterilization there can be two types of heat. One is dry heat and one is moist heat. ठीक है, एक तो ड्राई हीट के साथ स्टेरिलाइजेशन कर सकते हैं और इन वन वी यूज मॉइस्ट हीट. ड्राई हीट में क्या आता है? ड्राई हीट में कि वी heat the material or any article जो भी हमको sterilize करना है, we heat it so much that under flame that it gets red hot. ठीक है, and then we can also put it under flaming, उसमें flame से उसको heat कर सकते हैं. Then incineration and hot air oven. These are the four main methods of dry heat. For moist heat we have, it can be divided into three categories. We all know water boils at 100 degree Celsius. So the categories are, एक तो 100 degree Celsius से कम के टेम्परेचर पे अगर स्टेरिलाइजेशन हो रहा है तो एक कैटेगरी वो हो गई. दूसरी कैटेगरी है टेम्परेचर एट 100 और तीसरी कैटेगरी है टेम्परेचर अबव 100 डिग्री सेल्सियस. सो टेम्परेचर बिलो 100 डिग्री सेल्सियस इंक्लूड्स पास्चराइजेशन, इंस्पिसेशन, LTSF and water bath. Boiling temperature at 100 include boiling, tyndallization, Arnold sterilizer, and temperature above 100 degrees Celsius include one of the very important topics, that is autoclave. So autoclave is important theoretically also, and you all, if you'll be having your clinics, your nursing homes, then you have to keep an autoclave and you should know the function of autoclave. Autoclave से आप बच नहीं सकते, you should know the function of autoclave anyhow. So we'll discuss them one by one. Now moving on to the छोटे वाले पहले करते थे, तो short ones shall be discussed before. तो एक तो pasteurization. So we all have been studying it from our school days. What is pasteurization? Milk को हम pasteurize करते हैं. There are two methods. One is the Holder method and one is the flash method. In Holder method, the temperature is 63 degrees Celsius and it is done for 30 minutes. In flash method, temperature is increased, automatically the time period or the duration of exposure will decrease. In flash method, for a flash of 20 seconds, we sterilize it at 72 degrees Celsius, and then in both cases rapid cooling is done. So pasteurization, when we used to study in school days, यहाँ पर यही होता था कि पहले heat करो and then there is rapid cooling. So now at this stage, at MBBS or at UG level, you should know the exact temperature with time. And it is used to destroy non-sporing bacteria like Mycobacteria, Brucella and Salmonella from beverages and dairy products. There's one disadvantage, एक bacteria है Coxella, ये उसको नहीं destroy करता. So this is the disadvantage of pasteurization. Very importantly used in the milk industry. ठीक है, pasteurization of milk बहुत अच्छे से करा जाता है. So then the next word is inspisation. What is inspisation? Inspisation means fractional sterilization. Fractional means sterilization is done in fractions for 3 consecutive days. There is intermittent heating at 80 to 85 degrees Celsius. 80 to 85 डिग्री सेल्सियस पे 30 मिनट्स के लिए 3 दिनों तक लगातार करते हैं. Now what is the use of doing it for 3 days? See, what happens is that in the first day, all the viable microorganisms, let's suppose bacteria, all the viable bacteria is killed, but spores are still left. So what will happen the next day? The spores will germinate into bacteria. Next day you will kill those bacteria also. And then whatever spores again they are left, they will germinate, and on the third day we will again do inspisation, and all the germinated spores or bacteria, everything will be killed. Okay, and it is used for LJ media. LJ media, full form Lowenstein-Jensen media, and it is used for culture of Mycobacterium tuberculosis. And it is also done for Loeffler's serum slope. Okay. Now all media that we use in microbiology, that has to be taken for autoclave. That has to be sterilized by autoclave. But the temperature of autoclave is much higher. It is around 121 degrees Celsius. So those media that contain substances like egg or serum, जिनको इतना temperature पे sterilization नहीं चाहिए, for that we can go for inspissation, and it has to be done for 3 days. Okay, so two major sterilization techniques we have discussed till now: sterilization and pasteurization and inspissation. Then comes tyndallization. Tyndallization is also a form of intermittent sterilization. So what is the difference in tyndallization and inspissation? It is only of that of temperature. In inspissation, the temperature was 80 to 85 degrees Celsius. But in tyndallization, it is done at 100 degrees Celsius and for around 20 minutes, again for 3 consecutive days. The days should be consecutive. ये नहीं आज किया एक हफ्ते बाद नहीं, 3 consecutive days में ये करना है. It is similar to inspissation, only difference that is that it is done at 100 degrees Celsius. Again, the uses, it is used to sterilize so in sterilization of gelatin and egg, serum or sugar containing media. So this I have told you earlier also. So both these methods are similar except the temperature at which the sterilization is done. Okay. Now moving on to the to autoclave, which is the most important topic in this lecture. So autoclave, first of all, we should know what is the principle of autoclave. The principle is steam under pressure. So what is that? So we can compare the principle to that of a pressure cooker. The principle is similar to the pressure cooker. Pressure cooker everybody have in their homes. So what is the principle of a pressure cooker? So whenever it is a closed vessel, what happens when the water boils? Pressure also increases. With increase in pressure, the temperature at which the water boils also increases. Now, what is the boiling temperature of water? It is 100 degrees Celsius. But when the pressure will increase, the temperature at which the water will be boiling will also increase. So it will be more than 100 degrees Celsius. And once a temperature of more than 100 degrees Celsius is attained, then we can easily kill the bacteria, including spores. So let me read it for you. Water boils when its vapor pressure equals that of the surrounding atmosphere. So this is all physics. Water boils when the vapor pressure it equals that of surrounding atmosphere. Now when the atmospheric pressure is raised, the boiling temperature also raises. At normal pressure, what happens? Water boils at 100 degrees Celsius. Normally, पानी को उबालोगे, it will boil at 100 degrees Celsius. But when pressure inside a closed vessel increases, the temperature at which the water boils also increases. And moist heat destroys the microorganisms by irreversible coagulation, denaturation of enzymes and structural proteins. Okay, और we all know जो भी बर्न होता है स्टीम से ज़्यादा तेज बर्न होता है, तो स्टीम above 100 degrees Celsius can easily penetrate through porous surfaces. Okay. So this is a diagrammatic representation of autoclave, and here this is a picture of the autoclave in the laboratory. So we can see it is a closed container. ना बड़ा है, ये काफ़ी बड़ा है ना, and it is a closed container. और इसके अंदर क्या है? इसके अंदर नीचे जो है एक रेस्टिंग प्लेट है. Here you can see this is a resting plate. ना यहाँ पे resting plate है और उसके नीचे we add on water है ना. There is a level of water or till that level the water is added. Then there is a heating element. Heating element है that will provide heat to water and water will produce steam. उसके ऊपर एक बास्केट होती है ग्रिड की बनी हुई that has holes है ना. ग्रिड बास्केट जैसे छलनी जैसा, तो it has holes so that steam can easily penetrate. Or whatever material we have to sterilize is kept under the grid basket. Suppose we have to sterilize a culture media which is present in this beaker. So this is the culture media that is present in this beaker. Sorry for the bad drawing. So this is a beaker in which I have some culture media. It has to be sterilized. So I'll place it in the grid basket and then I'll close the lid. अब lid में क्या है? एक pressure gauge है that will show us the pressure. Then there is a safety valve, है ना, pressure बहुत ज़्यादा increase हो गया. Everything that a cooker has, cooker में भी तो ये होता है, safety valve होता है, pressure gauge नहीं होता, but there is a safety valve. Then air removal valve है और pressure control valve है और fastening screws हैं, है ना. ढक्कन खुल नहीं जाना चाहिए. Whenever the pressure will increase, the lid should not open. So there are it should be tightly closed with screws, and this is the lid. So now what happened कि ये सब इसमें दिख रहा है, प्रेशर गॉज है ना, यहाँ पे ये इसके स्क्रूज हैं लिड में, है ना, देखो कितने सारे क्लैम्प्स हैं, एंड दे हैव टू बी टाइटली स्क्रूड. देन वंस द वाटर स्टार्ट्स बॉइलिंग, हीट इज प्रोड्यूस्ड, इट प्रोड्यूसेस स्टीम, एंड स्टीम की वजह से क्या होता है, प्रेशर इंक्रीजेस, एटमॉस्फेरिक प्रेशर बढ़ा, देन द बॉइलिंग टेम्परेचर भी बढ़ जाता है, एंड द वाटर विल देन बॉयल एट 121 डिग्री सेल्सियस. ओके, सो इसमें मैं आगे दिखाती हूँ क्या होता है. सो देर आर 3 पैरामीटर्स विच आर वेरी इंपॉर्टेंट इन ऑटोक्लेव. वन, द टाइम पीरियड, कितना ड्यूरेशन टाइम पीरियड हमें देना है, और एक होता है प्रेशर, कितना प्रेशर होना चाहिए, और एक कितना टेम्परेचर होना चाहिए. ठीक, तो जस्ट स्लाइड. So procedure, as I have told you, material to be sterilized are placed inside the cylinder, inside that basket. Then steam jacket is filled with water, lid is closed, and the electric heater is put on. Sterilization process can be divided into 3 phases. First, सबसे पहले क्या होगा, the air is displaced by steam. Whatever air is present inside, it will be displaced by steam at 15 pounds per square inch. So this is the pressure, 15 pounds per square inch. Then there is a holding period. When this pressure is achieved, then we have to wait for 15 minutes. So this time period is known as holding period. And once this holding period gets ended, then the electric heater is switched off and allowed to cool. So the things that we have to remember in this slide is that autoclave is used at 121 degrees Celsius for 15 minutes at a pressure of 15 psi. So this line should not be forgotten by anyone. This is very important. The function of autoclave is at 121 degrees Celsius for 15 minutes at 15 psi pressure. Uses, it is used for all critical and semi-critical items which are heat and moisture resistant. Okay? For those items in which moisture will do no harm, they are moisture resistant, like surgical instruments, anesthetic equipment, dental instruments, OT gowns, etc. Then we can also use for culture media preparation, and for biomedical waste treatment also autoclave is used. Only the time period is increased. है ना, पहले हमारा time period था 15 minutes. In for biomedical waste treatment, it is done for 60 minutes for whole 1 hour. What are the precautions? It should not be used for sterilizing waterproof materials. ठीक है, जो waterproof materials हैं जिनमें वो steam penetrate ही नहीं करेगा, लाइक ऑयल है, ग्रीस है, या ड्राई मटेरियल्स हैं, उनके लिए नहीं यूज करना है. एंड द चैम्बर शुड नॉट बी ओवरफिल्ड, एंड सेपरेट स्टीम स्टेरिलाइजर्स और सेपरेट ऑटोक्लेव्स आर देयर फॉर बायोमेडिकल वेस्ट. ये नहीं है कि जिस ऑटोक्लेव में वी आर प्रिपेयरिंग कल्चर मीडिया, दैट ओनली वील यूज फॉर वेस्ट ऑल्सो. नो, सो देयर शुड बी सेपरेट. देन हाउ विल वी नो दैट आवर प्रोसीजर इज वैलिड और नॉट, है ना. सो फॉर दैट वी हैव स्टेरिलाइजेशन कंट्रोल्स. In sterilization controls, there are 2 types of controls: biological indicators and chemical indicators. So first, talking about chemical indicators. In chemical indicators, we have these autoclave tapes. Okay, these are autoclave tape-like material which have these brown strips. In these tapes, we have light brown strips. So we cut a small amount of tape and place it on the beaker. ठीक है, और फिर sterilization का process start हो जाता है. Then if your sterilization process is valid, then these brown lines are turned into black lines. These brown lines, they turn into black lines. Okay, if it is valid. और अगर ये change नहीं हो रहा है color, that means कुछ गड़बड़ है. Either temperature कम रहा या pressure कम रहा. ठीक है. And similar to autoclave tapes, you also have bovidic tapes. है ना, इसमें भी देखो पास में we'll see these brown lines, they have been changed into black. ठीक है, और यहाँ बीच में ब्राउन रह गया है, दैट मीन्स ये हमारा प्रोसीजर फेल हो गया. So these are all chemical indicators. Then comes biological indicators in which there is spores of Geobacillus stereothermophilus are used. ठीक है, we all know कि autoclave में we have to destroy spores also. ठीक है, तो the spores दिस इज अ फॉर्म ऑफ बायोलॉजिकल इंडिकेटर, है ना. ये बायोलॉजिकल इंडिकेटर की वायल्स होती हैं और हम अपने हर लोड के साथ एक वायल लगा लेते हैं और देन दीज वायल्स आर केप्ट इन इंक्यूबेटर. ठीक, इफ आवर स्टेरिलाइजेशन प्रोसेस इज वैलिड, spores would have been killed and geobacillus will not germinate on incubation. But if geobacillus germinates during incubation, that means our process is not valid, क्योंकि spores मरे ही नहीं, बैक्टीरिया ग्रो कर गया, है ना. So these are two types of indicators that has to be used during the process. Now moving on to next very important sterilization process, that is hot air oven. So autoclave was used in those materials in which that are moisture resistant. ठीक है, तो ऐसे मटेरियल्स जिनमें मॉइस्चर से कोई फर्क नहीं पड़ेगा, पर जो मटेरियल है जिसमें मॉइस्चर घुस ही नहीं सकता, है ना, इट इज नॉट पोरस टू मॉइस्चर, लाइक ग्लास हो गया. ठीक है, तो उसके लिए वी यूज हॉट एयर ओवन. सो यूज्ड फॉर मटेरियल्स दैट माइट बी डैमेज्ड by moist heat or that are impenetrable to moist heat, like glasswares, powders steam में करेंगे तो they will form liquid, है ना, वो destroy हो जाएंगे. petroleum products and sharp instruments, for these all instruments we use hot air oven. The procedure here is hot air oven. As you can see, this is a small almirah like structure and it is all made of metal. और ये इसका डोर होता है और ऊपर वही टेम्परेचर के नॉब्स वगैरह होते हैं. तो इट हैज़ अ स्टेरिलाइजेशन चैम्बर इलेक्ट्रिकली हीटेड एंड इनसाइड देयर इज अ फैन और अ मोटर टू इंश्योर एडिक्वेट और इवन डिस्ट्रीब्यूशन ऑफ हॉट एयर इन द चैम्बर. इसका टाइम पीरियड है 160 डिग्री सेल्सियस एट 2 hours. ठीक है, इफ वील इंक्रीज द टेम्परेचर, टाइम विल डिक्रीज. सो 180 डिग्री सेल्सियस फॉर 20 minutes or 30 minutes. Uses, it is used for glasswares like glass syringes, test tube, pipettes, for surgical instruments like forceps, scissors and scalpels, then for liquid paraffin, oil, powders that are used in lab. Again the diagrammatic representation, or ye there's an open hot air oven here also, so you can relate both the pictures. So this is almirah like structures, यहाँ पे बीच में slabs पड़े हुए हैं in which we can put our material. We have a thermometer पर है और अंदर की तरफ एक फैन होता है. Here also you can see there is a fan inside. Why is the fan used? Fan is used for even distribution of heat. Okay. So the advantages, it is not non-toxic and does not harm the environment, and it has very low operating cost and it penetrates well into the materials, non-corrosive. But the precautions are, again, it should not be overloaded and cotton plugs should be used to close the mouth of test tube and flask, and door should not be opened door should be opened only after some time when the hot air oven gets cooled. The biological indicator for this process is spores of Bacillus atrophius. So before moving on to filtration, let us revise about autoclave and hot air oven. In autoclave, the time temperature pressure was 121 degrees Celsius at 15 psi for 15 minutes, and it was used for culture media, for our all OT instruments. Okay, and in hot air oven, that was used for all glasswares, for sharps, for liquid paraffin, and for powders that are used in laboratory. And the time and temperature for hot air oven was 160 degrees Celsius for 2 hours or 180 degrees Celsius for 20 to 30 minutes. Okay, so whenever there will be a question, we have to write the principle. Then we also have to make a well-labeled diagram with this time, temperature, pressure. Uses, advantages, disadvantages can also be written. Then moving on to another method, that is filtration. Filtration acts by removing microorganisms, not by killing. Used for heat labile items. Okay, we have seen in autoclave and in hot air oven there is बहुत temperature जो है it is higher than 100 degrees Celsius. तो अब जिन those items that are heat labile, they get destroyed at a higher temperature, like vaccine, antibiotics, sugars and serum. Filtration method is used. So there are two types of filters, depth filters and membrane filters. Depth filters are used for drinking water purifiers, industrial application, etc. An example of this is seeds filter. I'll show you a photograph of seeds filter in my coming slides. Then there are membrane filters which are widely used in hospitals, which have a bacterial filter size of 0.22 micrometer pore size. Then filters, we have seen filters of, there are filters for liquid and then there are filters for air also. So you must have heard of this word HEPA filters that are used in OTs, to sterilize the environment of OT. So HEPA, the full form is high efficiency particulate air filters. It removes around 99.7% of particles that have a size of around 0.3 micrometer. Bacteria, they are very small, microorganisms are very small and their size is taken in micrometer. Then अब नए और आने लगे हैं ULPA filters. They are ultra low particulate penetration air. They have a lesser pore size. It is around 0.12 micrometer. Okay, and it removes around 99.9% of particles. And sterilization control for membrane filters is done by one bacteria, Serratia marcescens. So this bacteria produce pigmented colonies. And for filtration, it is used for sterilization of antibiotics and sugars that are heat labile, and separation of bacterial toxins and bacteriophages. So this is a diagram of seeds filter. Now the last physical method of sterilization, that is radiation. It includes sterilization by radiation, that can be cobalt-60 gamma rays in electron accelerators or infrared rays or ultraviolet rays. So low temperature sterilization method that has been used for a number of medical products. Okay, the pharmaceutical products like syringes, etc., they are all sterilized by radiation now. Medical devices, tissue for transplantation, etc. also. So radiation can be divided into two categories, ionizing radiation and non-ionizing radiation. Ionizing radiation causes the breakage of DNA without temperature rise. So what is the advantage? Ionizing radiation has a higher penetrating power and it is rapidly acting. When it will penetrate more, it will rapidly act. Temperature is not raised in this case, so it can be also known as cold sterilization. Temperature is not raised. The disadvantage is costly, okay, and may have deleterious effect on equipment made of polyethylene. For sterilization control, see, every process has to have a sterilization control. For this we use Bacillus pumilus. Non-ionizing radiation, it can be done either by infrared radiation or ultraviolet radiation, and infrared is the alternate method of sterilization for selected heat resistant instrument, but it does not penetrate glass. And ultraviolet radiation does not achieve sterilization, described under intermediate level disinfectant, and it is used to disinfect clean surfaces in OT, laminar airflow, etc. Okay, so ionizing radiation, again, these were X-rays, gamma rays, cosmic rays. They have high penetrating, rapid, and does not raise temperature. Again, therefore they are known as cold sterilization and used for disposable plastic supplies, catgut sutures, adhesive materials, and dressings. So that is all about the sterilization methods. Now we are also having some newer methods of sterilization. So you can just recall their names, like ethylene oxide. It is a form of gaseous sterilization. Then plasma pyrolysis, hydroclave, microwave. So these are all newer methods of sterilization, which the names can only be summarized. Thank you.